months, are you ready? Is there a delay, right? Are you ready? Cheers.
Testing. Good morning, everybody. Welcome to the first departmental grand rounds of the fall. A new season of grand rounds, and we are lucky to start off with a great one this morning. I'd like to make two quick announcements. First is to remind everybody that October 7th, which is just around the corner, we have the meet and greet at the faculty club. Hopefully everybody's RSVP'd, anybody who's a trainee, anybody who's a faculty member, or if you have any other affiliation with the department, please RSVP to that, and we'd love to see you there. And it's a great opportunity to meet new trainees. We always get the complaint that the only time our trainees meet each other is at graduation. And so this is a way to avoid that. It's the primacy-recency approach. We're going to have everybody meet each other at the beginning, if we can. And the second announcement is that after the grand rounds today, there is a session for trainees with our grand round speaker across the hall at 9:30. If you're rushing up to get questions with Mike after the talk and 9:30 comes around, you're out of luck. I'm grabbing him and stealing him across the side, but you can meet him over in the conference room across the hall with that. And with that, the most important thing I do in the morning at Grand Rounds is to introduce the introducer. Our introducer this morning is Jacques Wynn, who's a new faculty member, newly minted awardee of a K99, a highly prestigious award. And I'll hand the mic over.
Morning, everyone. My name is Jacques Wynn. I'm an assistant project scientist here in the Department of Psychiatry. I have the distinct honor of introducing today's Grand Rounds presenter, Dr. Michael Taff. Dr. Taff obtained his bachelor's degree from the Colorado College and went on to complete his doctoral studies in experimental psychology here at the University of California, San Diego. Following a brief postdoctoral stint in the Department of Psychiatry at UCSD, he joined the Scripps Research Institute as a postdoctoral fellow and was eventually appointed to faculty at TSRI in 2000, from which he has successfully continued to lead his independent lab. So just this past spring, 2019, Dr. Taff was recruited to join the faculty of the UCSD Department of Psychiatry as professor. So prior to joining the Taff lab nearly 5 years ago, I had a conversation with a senior investigator who I can recall aptly describing Dr. Taff as a PI who does it all. The work at the Taff lab focuses on the neuropharmacological effects of drugs of abuse and the resulting changes in behavior. His research program has investigated the potential harms and health risks that attend both acute and chronic exposure to recreational drugs, including MDMA, alcohol, cannabis, opioids, and the designer cathinone psychostimulants. His group has helped lead the field in the therapeutic development of anti-drug vaccines, and most recently, the Taff lab has helped pioneer preclinical models of inhaled drug delivery using electronic cigarette technology. With over 25 years of experience in vivo neuropharmacology, Dr. Taff has been the recipient of several awards from the National Institutes of Health, has published numerous original research articles, editorials, and reviews. His exemplary commitment to a professional and academic service has included serving as member of editorial boards, service committees, and professional societies. However, most notably, for 20 years, Dr. Taff has served as a dedicated mentor and advisor to numerous postdoctoral fellows, graduate and undergraduate trainees. He's someone who truly is an innovative leader within the field of drug abuse research, a devoted colleague, and a great mentor. So he educates, he instructs, and he inspires. So in other words, he does it all. It has been the greatest privilege of my career to be a member of his team, and now together as members of the UCSD Department of Psychiatry. So again, it is with tremendous honor that I introduce to you today's Grand Rounds presenter, Dr. Michael Taff.
Thank you, Jacques. Another way of saying I do it all is I'm a bit of a dilettante. So I have no conflicts of interest with this presentation. I want to start with the acknowledgments because a lot of the work I'm going to talk about is Jacques' work in combination with Yannabelle Grant, technician in my lab, and Kevin Crean. I'm going to then discuss some collaborative work with the Janda Laboratory from TSRI, and Candy was the main postdoc collaborative, Jacques, on that thing. And then Dean Kersen and Marissa Roberto's lab was a collaborator. Some other work that I'm going to talk about. And then finally, of course, NIDA for funding. These are the awards that funded basically the work I'm going to talk about today. So some smart aleck a little while ago tutted his academic society and suggested that Instead of concentrating so much, well, I didn't say we shouldn't concentrate on treatments and therapies and mechanisms, but I did point out that I think the primary translational product that we generate in drug abuse science is information. This is the thing that has the greatest effect on the drug abuse crisis. And you can think about this, there's numerous anecdotes, but the ones I like to point to are cigarettes, right? So cigarettes, the older people in the room know this, the younger may not, but we were still fighting deep into the 90s about the idea that cigarettes were indeed addictive, and proving that they were addictive and showing that they were addictive throughout the past century has had the greatest effect. Cocaine, this is a really old one for some of you, but back in the 80s, cocaine was safe and wonderful and business people were supposed to take it. There was a death, a heart attack death of a basketball player. This was no news to medical science, people knew this, but it reached a lot of people. The information got out, and so the perceptions of cocaine safety went precipitously down in 1 year, and use rates went way down. I've got a history of interest in MDMA and ecstasy, or ecstasy. This is a population which is exquisitely sensitive to the science, or has been, at least through the 80s. And so this is an area where things that people were doing to try to reduce their harm, to recognize harms, came out of research findings. And there was a California medical marijuana initiative that we passed a couple years ago where you could, when I was reading through it initially and deciding how I was going to vote, there were a lot of things in there that were formed by existing science. And there were a lot of things in there that you said to yourself, we could use some more science to inform what we should do in terms of policy going forward. And that's still very much true. As you know from Igor's and Tom's work. So one thing that informs my lab and my approach to preclinical science that I think it's really important for us to generate information that can be actionable for public policy, for medical decision making, for parental decision making. I'm a parent of 3 kids, teenagers in the critical zone, and individual decision making for you and I. So what about the opioid crisis? These epidemiology slides you've probably seen or may not have, but this is one of the first ones that was trying to get people's attention that as motor vehicle deaths have been declining in past years, we had a crossover point, 2008 or so, where all of a sudden drug-associated deaths were more frequent in the United States than car accidents deaths were. Most of this is opioids, not exclusively, but a lot of this is opioid related. It's been coming for a long time. This is, for those of you who can't see, this is 2000, this is 2008, this is 2016. Natural and semi-synthetic opiates, this basically means treatment drugs. Oxycodone, hydrocodone, and the like. You can see deaths associated with this drug increasing past decade and a half, if not 2 decades by now. More recently, you have an uptick in heroin deaths. And then in the last few years, this is what's actually brought it to people's attention. This is the fentanyl crisis. This is highly potent, another synthetic, but the appearance of deaths associated with fentanyl has been what's brought this to the news. I'll go through this backwards because I want to get to this. So if these prescription opioid deaths have been increasing since 2000, we've known all about this epidemiology, we've known about the use of opioids, why did self-administration, but preclinical data lag, and preclinical data did lag. So this is a very simple search of PubMed for self-administration. It's a primary rodent model and non-human primate model, and either heroin or oxycodone. And what you can see is that there's plenty of papers and plenty of work studying the effects of heroin in preclinical models. And what happened is that there's essentially nothing, very few papers published on oxycodone until maybe the last half decade when you start to see an uptake. And you're going to see a big uptake next year because things have been picking up. But you know, the crisis has been developing this long. Why did it take so long? Because we fail. Not everybody has my attitude, basically. Okay, so I want to address, one thing about the opioid crisis. I'm not going to talk about data that directly address these things, or not necessarily, but I do want to do another little bit of tut-tutting here, which is that there, currently are kind of 2 main theories about what's really driving this crisis. Why are there so many people using opioids than before? There's the Brett Favre hypothesis. People who are treated for pain conditions and just continue using it and get non-medical use and become addicted. And there's the availability hypothesis, which is basically that there's a ready supply because of broad prescription for oxycodone and hydrocodone and the like. They sit around in grandma and grandpa's and parents' medicine cabinets, and they get diverted, presumably and primarily by adolescents who are the target population for drug abuse. All right, so there's evidence for both these things. This was a 2017 report. This basically shows the probability of continuing use. Goes to about 50% here, if you can't see that, given how many days supply your first opioid prescription was. So if you only had 5 days, you're very unlikely to continue using in a year. If you were given a month or more, almost half of you were continuing to use opioids a year later. And so this is kind of the one of the pieces of evidence we have that maybe there's something to this continued use after prescription, that we can do something in policy-wise about how we handle that. The availability thing, this is just a study of adolescents and why they use prescription opioids. They were not sports kids with injuries. They were just kids that got access to diverted medications. So both these things have support, and both these things are things that could be amenable to, or at least primarily the pain one, could be more amenable to preclinical research that we're really not doing a very good job at in terms of looking at addiction in pain models. But it's coming. Okay, so today I'm going to talk to you about three main things. We're going to talk a little bit about vaccines. Are they an answer to the opioid crisis and more generally the drug abuse crisis? We're going to talk about THC or cannabis. And then we'll talk a little bit about some theoretical underpinnings and what some of our studies have suggested about these constructs that have been promulgated here in the West Coast, particularly San Diego, by drug abuse scientists, most primarily George Koob. So first I'll talk about vaccination. So Jacques collaborated with Candy and the Janda laboratory. Not to insult your intelligence, but just going to talk an awful lot about self-administration today. So many of you know you can prepare a rat, a mouse, or a monkey experimental subject with an intravenous catheter line that has an externalized port. You hook it up to a pump which pushes small infusions of drug, made contingent upon a response of the animal, in this case, this lever. Basic operant psychology stuff. This is the backbone of self-administration. You can do all kinds of things with this model. You can determine drug potencies. Sort of what doses are necessary to reinforce. You can determine something about efficacy. Conventionally, how hard will they work for a drug? This is a question you can ask. Will they self-administer at all? So, sort of famously, rats won't self-administer THC and LSD. MDMA is kind of a maybe. And then you get into fancier models. So I'm going to talk a lot about the escalated model of as an improved model, I guess, of compulsive drug use. And then you can talk about relapse. So do people go back to using, their animals go back to using after a long period of abstinence? I'm not going to talk about that particular model. But I did want to just kind of overview this. I'm going to talk a lot about this. So are oxycodone vaccines the answer? For those of you that haven't heard about vaccination against drug abuse, it's really super simple in concept. It's dose reduction. The idea is you generate antibodies that recognize the drug molecules. They bind them in the periphery, in the bloodstream, keep it out of the brain. Therefore, you reduce the effect that's getting to the dose that's getting to the brain. Drug small molecules do not generate immune responses by themselves, so what you have to do is couple something that looks like the drug or the drug itself, in this case, to an immunogenic protein, in this case, KLH or keyhole lipid hemocyanin. The idea is you vaccinate, the body generates antibodies that recognize all kinds of pieces of this, including a lot that recognize the drug-looking part of it. And this is an example for cocaine, but basically you have antibodies that recognize now the drug molecule that wouldn't have been present before you vaccinated them, and keep it in the periphery. We have a long history showing that this works, and I say works in quotes because we have biological efficacy and we have what you might think of as psychological efficacy. So this started in '74. There was a big gap after this for reasons I'm not really clear on until about '95 when Koob and Janda started publishing on cocaine vaccines, and Carrera was a grad student with me in psychology at the time, showing that you had efficacy in behavioral models. And this led to essentially an uninterrupted cottage industry of study on vaccines against drug abuse. It has led to at least 2 clinical trials, a nicotine one and a cocaine one. I'm just going to give you a little overview of why these are considered to have failed and why we're continuing to work on this question and what we think we can contribute. So the first thing is that you vaccinate, you have a vaccine schedule, you have a vaccine you've identified in preclinical trials, you give it to people, and these are antibody responses of all the individual subjects. Across 12 weeks, 16 weeks, you discontinue vaccinating, they kind of subside 24 weeks. But these are weeks, they're developing this antibody response, and there's incredible variation, as you can see, across individuals, right? So a lot of them are down here, there's a few that go like this. This turns out to be important. So, sorry. Why was it negative? Well, it was negative because there was no group effect between the placebo and the vaccine in terms of cocaine negative urines. This was a population that was dual cocaine opioid dependent. They're in methadone maintenance and cessation therapy for the opioid. And I don't think they were particularly under cessation for their cocaine use, but they were dual cocaine users. They were vaccinated, and they were assessed for their percent cocaine-free urine. So up here is good. This is the placebo and one of the groups. And there was this one interval of time when some of the group, and what they did is basically they cut off this group that had a high antibody response, said, aha, it's successful. So you can't really do this in clinical trials, just declare them victories. But it was kind of a victory in the sense that, you know, if you gave a small molecule trial and most of your people didn't take the drug or didn't achieve drug levels, you wouldn't really consider that to be a failure, right? So the same kind of thing goes into these trials, but they're super expensive. There's not a lot of will when the general thing is considered to be a failure. Same thing happened with the nicotine vaccine. These are the group mean effects. You cut off the top 30% of antibody responders, and all of a sudden it looks like they're effective. We do the same thing basically in the rats. So we vaccinate them. That's with the, multiple challenges here that then bring up antibody titers, 2 different cohorts. They have we have variation. And then once they get to stability, you can start doing behavioral tests. And eventually they'll subside despite your best attempts. I'm going to show you first some intravenous I'm going to show you both intravenous self-administration and nociception assays. But first thing is does it biologically work? This is what I was telling you at the beginning. This is just showing in rats that if you give a challenge dose of oxycodone, the positively vaccinated animals have retained a lot of drug in their periphery compared to the controls. If you look at a blow up here in the controls, you can see this dose and time dependent. So that's 15 minutes and 30 minutes after the 1 milligram per kilogram dose, and this is the 2 milligram per kilogram dose. So here, same time point, you see essentially a doubling of the ratio, so it's dose dependent. You see again that this group retains a lot of drug in the periphery, but importantly, you get this sort of lack of linearity in the dose effect here in these 2 bars, as if you've sort of reached a saturation point. The antibodies can no longer suck up drug after a certain dose. And then, of course, more importantly, the periphery is in the brain. So this inverts. The controls have more than the vaccinated animals. This has functional effects. This is a nociception assay. So basically, you put the animal's tail in hot water and it flicks it out within about 3 or 4 seconds under control conditions. You give a dose of an analgesic. And they slow down their removal rate. These are the controls because they have an analgesic effect. This is attenuated in the vaccinated animals, but you can dose past it by doubling the dose to 2 milligrams per kilogram, and all of a sudden you pick up more of the response in the vaccinated animals. So again, it's kind of the same idea, which you can saturate the effect. It's a partial effect. Okay, so does it work for self-administration? And the answer is at first no. So this is very reminiscent of the human stuff. We look at group mean effects and you say, well, it didn't work. They take the same amount of drug. We vaccinated them. We started training self-administration. These infusions earned across 18 sessions. But if you break the population down into a median split, so top half, bottom half, the ones in the middle here are the controls. They're very tight. The population is very similar. But you get this bimodal distribution of the vaccinated population with some taking very little and some taking even more. Well, if they had a reinforcing effect, this is what they should do, right? Because you've decreased the amount of the doses getting to the brain, so you might expect them to compensate, although they fail to fully compensate. Another way to look at this is the percentage of the animals that reach acquisition criteria. So if you establish one based on some things of knowing about the drug, you see that eventually all the control animals meet criteria for self-administering. They're very tight. About 40% of the animals fail to meet some arbitrary criteria for learning this discrimination. Is this important? It's kind of important. We often forget with preclinical models, because we want to test all the animals doing essentially the expected phenotype, is that dependence is a minority outcome. It's a severely minority outcome for every single drug. So what this is heroin. I'm going to point you to. This is given that you tried a drug, are you still using it a year later? 70% of people aren't for heroin, and only 13% of those people who have tried heroin are dependent a year later. And this is the same for all the drugs. The only real differences are that your chances of still using non-dependently for alcohol and marijuana are actually higher than your chances of not, which is a little bit unusual for these drugs. That's the same slide I just showed you. I wanted to point out that it works for methamphetamine vaccines. There's the same kind of idea here, where you're looking at an acquisition criteria. You go out to 8 sessions of methamphetamine, you get this big gap, and eventually they'll catch up. But you got to remember, people don't just keep taking meth until they get addicted, right? There's tons and tons of those people who are in that minority that say, oh, I tried pot or whatever. You were encouraged to try in college, and it just didn't do anything for me. I didn't take it. So the question is, can prophylactic vaccination be used to prevent acquisition? As you can probably imagine, the surest way to prevent a drug abuse problem is to prevent it from getting established in the first place, right? So prophylaxis is a good model, or is a good target, because you can make that minority outcome even less common. I'm a Johnny-come-lately to this topic. And I do all these things that they say, well, you can't do that. You should not have done these studies because prophylaxis will never be the target. My approach is that we're preclinical science. Why should we not do the studies? Because we think it won't be approvable, we think it won't be acceptable to people until we show whether it works. So we do it because we see whether it works. And now we can ask the question about, should we do it, right? And I kind of think the HPV vaccine for adolescents has kind of answered a lot of these complaints. We've already decided in one case that we're willing to vaccinate our adolescents on a broad population basis to avert some health crisis. So I think it's worth discussing. Okay. This is a second thing which has to do with the failure of human clinical trials. So this is a slightly different cohort of vaccinated animals. I'm going to point you to, this is basically terminal training conditions for the controls and the vaccinated animals I showed you before. There are some that will take more. In this case, because we had a higher training dose, more of the population took more. So under easy access conditions in FR1, one press, one infusion, vaccinated animals take more, but they don't fully compensate for what we infer to be their brain load. If you increase the workload in the controls, they'll defend against it. So this is a progressive ratio procedure where they have to make more responses for each subsequent infusion of drug. There are two different steepnesses to the schedule here, which these two bars are. The controls basically defend their intake against this. The vaccinated animals don't, and so they'll really decline, and they're more labile to challenge, to increase workload in this case. And again, you go back to the human trials, most of them are dealing with how much drug is being taken in. Cocaine-free urines, smoking point prevalence of abstinence. Well, you could have a situation where individuals are taking drug, maybe even taking more drug to compensate, but they're undercompensating, and this could have a protective effect. And this is an area where we've got to establish some preclinical models so that it can feed forward and say, well, we need to design the clinical trials to respect this, right? They might be less addicted despite the fact they're taking more drug. All right. That's it on vaccines. It works. Acquisition can be prevented, so we probably talked about prophylaxis. And then the question is maybe we can give more endpoints that we should be looking at in terms of human clinical trials based on what we learned from animal models. I turn to THC. Our work is kind of threaded by three main observations. That one, there's a sort of a reduction of opioid harms, including overdose in medical marijuana states or recreational states. There's some anecdotal and more formal data suggesting that marijuana can enhance pain control of analgesics. And then there are some proposals that maybe smoking weed will help people reduce their opioid use for addicts. All right, so about the vapor. So a few years ago, sort of the old guys on my hockey team who are, they're San Diegans, they've had their weed game going for decades, and they've switched over to vape. So I was like, okay, well, if these guys are switching over, it's really something. So I'm already in my lab, collaborated with Maury Cole, as some of you know, to develop a vapor inhalation model. It's just a sealed rat cage. You have e-cigarette cartridges, and you can sort of fill the chamber with THC vapor and other things. It produces THC typical effects on thermoregulation, anti-nociception, and other measures. Okay, this is an example of it working for this tail withdrawal assay, so warm water assay. Again, they flick their tails out in 3 to 4 or 5 seconds in this case. It's very stable across time. This is 2 hours after vapor exposure. If you vapor expose them to THC or oxycodone alone, you have this partial effect at these doses. If you give the combination, you have basically an additive effect. So this is the thing about these drugs acting together potentially for a therapeutic purpose. And it works in our model, which then brings me to the self-administration data. These are now 8-hour sessions. 8-hour sessions, which I'll get to in a while we start to stretch these out. But this is the number of awards during acquisition of self-administration. Animals go up and they kind of stabilize after a while. You can start doing interventions, treatment interventions. So if animals are exposed to vapor that contains THC before the session, compared to a session where they've been exposed to the vehicle, the PG in this case, it changes their behavior. So these are infusions not across sessions, but across the 8 hours of the single session. And they're very stable under vehicle conditions. And we get a reduction with THC in the amount of oxycodone they're taking. It lasts up to 3, 4, 5 hours. It's dose dependent, so this is a replication here. That same dose, you half the concentration in the vapor, and you have a partial effect. Again, it lasts for hours. So that's pretty cool. This goes to this question about whether or not people could be smoking or vaping, in this case, marijuana extracts, to have a protective effect. The first complaint anybody has is, oh, it's just THC, it suppresses behavior, the rats won't respond. We have some reasons to think it's not the case. I'm going to point you down here first. These are basically two different replicate cohorts. I'm not going to talk about what the groups are, but you can think of them as two replicates because the group thing didn't work. But basically, under vehicle pretreatment conditions, depending on what oxycodone dose you give them, they will compensate for a reduced dose or compensate for increased dose, depending on how you look at it. So this is the higher concentration. This is the lower one. They'll take more infusions at the lower one. We've switched to injection of THC here to get better dose control, tighter dose control. So these are milligram per kilogram IP doses. You have this linear reduction, this parallel reduction in how much oxycodone they take. This is important, right? So if it was just a general suppressive effect, they should just kind of stop their behavior, right? It should be the same. There shouldn't be any implication for what dose of oxycodone they were getting, but it does. And that's the first clue that it's a synergistic effect or an additive effect where you're actually increasing the efficacy of a unit dose of oxycodone. Most importantly, so this wasn't exactly the cleanest thing. This is a fixed ratio dose substitution. We switched this group to heroin. So this is heroin self-administration in this case, and this is a progressive ratio. So the good thing about the progressive ratio is that you can get different estimates about where in the dose curve animals are willing to work hardest. So the potency relationships can be a little different between FR and PR curves. But what we're able to see with this is this is a vehicle pretreatment now showing you breakpoints instead of infusions. So this is how many responses the animal would make for the final infusion of drug. And if you give a low dose of THC, which didn't do anything under FR, it actually increases how hard they'll work for that single fixed unit dose of heroin in this case. It increases the efficacy of heroin. So that's a case where you can't say that it's just suppressing their behavior. It's not just reducing their willingness to work or their ability to work. They're actually working harder to get that unit dose. So our conclusion is that it's the THC is increasing the reinforced efficacy. It's not a replacement. There are reasons to think this from pharmacology because we know that this endocannabinoid system is a modulatory system that regulates the function of many synapses, including opioid ones, so it makes sense pharmacologically. So the real question from this is, and we aren't onto this yet, is can it work as therapy? So the question is that a protective effect? You take a combination of drugs, it lowers the amount of each drug individually that you need to produce the same effect, in this case a reinforcing effect, but it could be the pain control effect. The amount of drug that's experienced is related to the plasticity, which is related to some of the negative outcomes, like addiction. So it could be the case that this is great, because you reduce the amount of opioids somebody's taking, less plasticity, less chronic exposure, you have a beneficial effect. Or it could be because it's modulatory, you're basically getting the same effect on downstream signaling, and you're not getting anywhere, because you're giving them the same effect. And so those are things that need to be worked on in the future. Okay. I'm going to turn to more compulsive models and some of the more theoretical aspects of the work. As many of you have seen this slide ad nauseum, this is a cube figure. It reminds us that there are several aspects of the drug abuse cycle, including the actual intoxication phase, the preoccupation or anticipation, and the withdrawal and negative affect. And of course, most of you are aware that this area of the world in drug abuse research concentrates on this negative affect, this withdrawal, and the idea that it's negative reinforcement that's driving continued drug use. The idea is, for those of you who forgot your basic psychology, is that it's the removal of an aversive stimulus that's driving the behavior, that's reinforcing and increasing the probability of behavior, as opposed to the feel-good, the provision of an euphoric stimulus. Okay, so you've seen versions of this before too. The idea is that drugs have potentially a euphoric phase, which then subsides at some time on some time course. There's then a dysphoric or a hangover phase, which then subsides on some time course. And the idea is that with repeated cyclings of this A, so-called euphoric process, and the B, the dysphoric process, you may get a situation where there's two different ideas. One, that the peak of the euphoria process kind of gets blunted, they can't get as high, and the trough of the negative process gets more exaggerated. Then there's the idea that maybe you have this kind of progression downward where not only are you blunting your euphoria, but your euphoria starts from a hole in the ground, right? You start from a point of dysphoria, and you can't get back up even to normal. This was modeled by Serge Ahmed years ago now by switching from short access models. So most people had done self-administration in 1 or 2 hour sessions because it was convenient, you had limited box time, and it worked fine. to ask about the A process of euphoria. And if you do that in short access 1 hour sessions, animals will take very, very stable amounts of drugs session to session to session for cocaine and other drugs. Serge's innovation was that if you give animals 6 hours of access to cocaine, a stimulant, what you get is a progressive increase in intake. And this wasn't just for, as you might imagine, 6 hours, they have more opportunity to take it, but they actually increased their intake even in that first hour. They hit it harder even in the first hour compared to the 1 hour group. This works for some drugs, but not others. I kind of alluded to the fact that MDMA doesn't really get self-administered terribly well by rats, and so they don't tend to escalate under 6 hour conditions, as data from my lab. But a very closely related cathinone, methylone, which looks exactly like MDMA, except for it's distinguished by the thing that distinguishes amphetamines from cathinones, they will escalate. So very closely related drugs can result in very different behavior. And of course, for the purpose of this talk, this goes back to Serge's work with John Walker. This is a heroin study. So this is long access to heroin compared to short access to heroin, and they escalate. So getting back to that figure about our failure to look at self-administration of oxycodone until very late in the game, the question is there anything special about these drugs? Like, is it just functionally another opioid and it's all about access and all that? Or is there something different about this particular drug in animal models? That's to remind you of that figure. And then right at the end of George's tenure here, before he went to the NIAAA, Kerry Wade had shown that, yes, indeed, behavioral responding for oxycodone in long access conditions will escalate compared to short access conditions. So we kind of took up the project from there. This is work from Jacques. So the first thing to do is to kind of just recapitulate that idea, whether or not if you give the animals long access, in this case 12 hours versus 1 hour, do they tend to increase versus staying very stable? That indeed happens. And we notice something very strange, which you might have picked up on. It's not very smooth. So what are these? Well, these are weekend breaks. Being the nice mentor that I am, I tend to run a 5-day-a-week lab, and we tend to take weekends off. And so what this is this is a 60-hour discontinuation as opposed to the usual 12-hour discontinuation. What we thought was we got a novel test of this hypothesis on our hands. So one possible explanation for that is that, again, if you think about this A and B process, you might be introducing the re-access to drugs anywhere in this slide or anywhere in this recovery. And if you do it in the trough, right here, you might have a harder time to get back up to normal. The animal might take more drug. As opposed to if you happen to do it here, yeah, they'll need to take drug to get back up to normal, but they have to take less. So we thought, well, this is an interesting way to test the hypothesis, which hasn't really been tested before. And then what happens if you interfere with this development of dysphoria? So can you manipulate where you are in this process and then give them access to drug? So this is a replicate cohort showing that same weekend stair-stepping effect. And the first thing Jacques did was say, what if we eliminate those breaks and run them 7 days a week, right? 12-hour access, 7 days a week, heroic work for him and the technician. You basically eliminate that stair-stepping. They'll still drift upwards, but you eliminate the stair-step by just preventing those 60-hour breaks. This is now a control that is saline self-administration. It'll come up in a minute why I'm pointing this out to you. It's not the short access, it's saline self-administration for the same 12-hour sessions. So why is it a key test? The negative reinforcement explanation kind of predicts that there should be greater effect, better reinforcing effect when dysphoria is maximized. We know dysphoria waxes and wanes with the interval. And prior models have not incorporated this in explicit test. What they've done is they've kind of selected intervals based on maximizing drug take for a given compound, but they haven't really tested this explicitly. And then I'll talk to you about, again, interfering with this dysphoria, why we're interested in the kappa opioid signaling, with dynorphins being the endogenous ligand for the kappa opioid receptor. And does this contribute? And again, this is a collaboration of Jacques with Dean in Marissa Roberto's lab to look at some cellular electrophysiology. So this is just a reminder slide, two different things. Again, you've probably seen these from Koob, both the central nucleus of the amygdala, so we think of this as the negative affective side, the dysphoric side, the stress side. Kappa dynorphin is represented here. And the more reinforcing side, the sort of nucleus accumbens projection, the dopamine responses, those are also places where the dynorphin kappa signaling is regulating function. There's a great tool for behavioral pharmacology. It's almost magic. It's called NorBNI. It's a kappa ligand, and it apparently inactivates the kappa opioid receptor signaling for something on the order of weeks after a single systemic injection. That's great for something like self-administration. So it's been shown to work in other escalation models. This is data from Joel Schlossberg with George, showing that if you have heroin extended access, giving a pretreatment of this NorBNI compound blunts the escalation. It doesn't affect short access, 1 hour sessions. These are summarized data here. It works for methamphetamine. There's nothing magic about the intervals. This is now a 6 hour thing for stimulant, 6 hour long access. You give a pretreatment of this NorBNI compound. It blocks the escalation. This is, again, summary data. Doesn't work for short access. So the obvious question is, could we use this to test our idea that what was important was this 60-hour discontinuation that had to do with the development of dysphoria? So these are the same data except for the red one, and this is where animals had 5 days on, 2 days off, just like this group, except they had pretreatment of this NorBNI compound. And sure enough, it turned them into a group that looked like their intake looked like the 7 days a week, as if we'd bridged the 60 hours or, you know, diminished the effects of that 60-hour discontinuation. Cool. There are electrophysiological correlates of this. So again, this is Marisa Roberto's lab, and she's very interested in the function of GABAergic cells in the central nucleus of the amygdala. These are electrophysiological recordings. Our approach to this, and Jacques and Dean's approach to this, was formed by a finding from Marcita Kalupi, who's now an assistant adjunct professor in psychiatry now. But what she'd shown Marisa Roberto a while ago is that in extended access cocaine self-administration, let's get a little nerdy, but these cells, these GABAergic cells in the central nucleus, if you're looking at mini inhibitory postsynaptic currents, this gets a little confusing, superfusing on the kappa antagonist onto the slice. Okay, so these are just, they hadn't been pre-treated, they were just sort of extended access. And compared to naive animals, you superfuse on this antagonist, it increases the frequency of these responses. And in the long access animals, that's absent and they actually go down compared to naive controls. This is the experiment. I'm just going to draw your attention to this because it's the parallel to this figure. So the first thing that Jacques and Dean showed was that again, it's a saline group instead of the naive controls in this case. I pointed that out to you. Short access versus long access, and you get kind of that same pattern, which is that the saline animals look like naive controls here, and the long access animals, again, oxycodone 12 hours as opposed to cocaine in 6 hours, they look the same, right? You have a decrease in the frequency when you superfuse on NorBNI. Very similar to these prior findings. What about the other groups, the critical to test groups here, the 7-day runs and the ones that were pre-treated with NorBNI? Again, this is the similar part of the figure, the same manipulation of superfusing on the kappa antagonist onto the slice in these different groups. The black one is the one I just showed you, that the response is to decrease the frequency of these mini inhibitory postsynaptic currents. And what you see with either the 7-day or the 5-day on, 2-day off NorBNI pre-treated animals, you see this normalization back towards the baseline as if they were short access animals. They don't look like saline controls, they don't look like the naive animals from Marcita's work, but they are normalized in their response back towards what the short access animals look like. So again, some evidence, we have this kind of accumulating evidence that this is dysphoria, things that cause and mediate dysphoria in long access drug models. They seem to be identifying that 60-hour discontinuation as something important. So we want to do another additional test of this dysphoria hypothesis. Most of you will be familiar with the intracranial self-stimulation reward model that Athena Marcoux pioneered and applied in a lot of early studies of this escalated compulsive long access work. The idea, just briefly, is that you compare animals with an electrode-directed medial forebrain bundle. It has a reinforcing effect in operant paradigms. But the idea of this particular approach is to determine the threshold of detection for how much current it takes for that animal to detect a stimulus. And basically, when it's well trained, it's sort of over here. I'll direct your attention here that you present a stimulus. It has to make a response within 7.5 seconds to say, I felt that, and then it gets another one. They're trained not to respond if they don't feel anything with timeout punishments. And what you can do by raising the current up and down, you can determine a threshold for where that animal likely is detecting current, where it detects a reinforcing effect. It's been considered to be a non-pharmacologic, if you will, assessment of where the brain reward, how euphoric or how dysphoric an animal is. If you give ascending doses of cocaine, you decrease these thresholds, less current required, they're pro-reward. This is data from my laboratory showing you get the same effect with methamphetamine, whether delivered by vape or IP injection, same thing for a couple of cathinones. You give an acute drug injection to animals that are otherwise drug naive, they feel good, decreases the threshold. The same thing happens for discontinuation from dependency situations. These are alcohol data from Gary Schultheis, Athena, Maury Cole, and George Koob, showing that if you discontinue animals from alcohol, there will be this time course dependent elevation in these thresholds as if they're dysphoric, which will eventually resolve back towards the baseline. So you can kind of track those cycles that I was talking about, the A and the B process with this procedure. And sure enough, long access to heroin, so this is back in the day, this is Athena and Paul, Kenny and George, showing that in animals that are escalating their intake of heroin because they have 23-hour access, their reward thresholds are also being elevated when tested right after the session, which is interesting because they're actually tested after the session when they could be acutely intoxicated. They're still driving the reward thresholds up. The other thing to point out is just in the short access animals, if you record them pre or post heroin availability, having heroin on board will decrease their thresholds, much like those acute injections. So it's self-administration of a drug versus investigator-administered drug. So Jacques and Janabel prepared some more animals for the self-administration study after they've been trained to do this ICSS procedure. So they're giving us a readout for their brain reward thresholds, very stable by the end of training. Then we start self-administration. This is basically a replicate of the infusion pattern for long access and short access. You see these same step-wise breaks. I'll tell you in a minute about what was different about this. We omitted the self-administration on a Thursday. This is a summation of those Friday, Monday junctures in the long access and the short access animals. So we looked at the reward thresholds, and sure enough, just like the prior data, in the first week, as they're establishing their self-administration, up go their thresholds. Beautiful. Critical test, right? What about that 60-hour interval? Uh-oh. They reset, and then across the week they went up again, and here's another week. And this is the day I was telling you about before where we omitted after this test. So these are all recorded before the self-administration session that we give them their 11 hours in this case, and then 12 hours discontinuation to record them again. So this would be before that Thursday session. We omitted the self-administration, and within that 36-hour now interval, they're already starting to come back to this baseline. So it's fairly linear. Right? It's not like there was something funny going on in that 60 hours. It looks like it's fairly linear. Okay, well, and I have to tell you, we don't know the answer to this yet. It is paradox, and we're not entirely sure how to explain this because we really expected that to go up. So what have we done to sort of look at this? So one, sort of a little bit of a brief diversion back to the thing. We looked at this critical NORBNI test in the same model. And what you get is, so this is the saline group, so this is the controls, basically. Get that same resetting upwards, so there's a replication. Maybe it's not as complete of a reset. And then we have the NORBNI pretreatment, right? So this is the one where the intakes were stabilized. They look more like the 7-day run animals. And it, at least in a group mean basis, and some of these individual points come out, you see an attenuation of this threshold elevation, right? So they're not getting as dysphoric, despite the fact they're doing the same self-administration for 11 hours. All right, so maybe part of this is not really answered, but another part is, right? It's consistent with the NORBNI effect. You see this in the way reward thresholds are modulated. These are the same data I just showed you. It's basically the first cohort and then that saline group. So this is basically a replication. So they overlap on each other. And then if you look at the short access from the first study with the NORBNI treated long access from the second study and overlap those curves, what you get is you see this intense impression that what you've done is you've normalized the long access animals back towards the short access animals, which should be reminiscent of what I showed you from electrophysiology, right? That you don't get a complete recovery. You don't have normal animals, but you've taken these long access self-administering animals and you've normalized them back towards what looks like a short access phenotype. Yeah, and that's what I'm just reminding you here of what I just said. It's normalizing it back to a short access phenotype. Okay, so in terms of the paradox, and we're kind of out a little bit speculatively here. So I showed you those acute data. So if you take naive animals, you give them an acute dose of most reinforcing drugs, stimulants, opioids, you will get a reduction in their thresholds. So in the first cohort, we did an experiment where on one of those Mondays, we recorded them as normal. We let them have only 1 hour of self-administration of drug, and then we recorded them again to find out if you could basically if they could treat themselves by self-administering drug. Didn't budge on the Monday. Again, by the time you get to the Friday, their baselines, their first recording of the day has been elevated. I showed you that already. Now, if you give them access to drug, it does reduce reward thresholds, as if you're sort of feeling less dysphoric. That's the kind of thing that's evidence that that's what's driving self-administration. Is they feel less dysphoric, they're treating themselves. But it didn't work on the Monday. Okay. This is the second cohort. This is sorry, I didn't say the saline, but this is actually the saline group. These are now just the end of the weeks that we're trying them because that's the only thing that worked in that first study. So these are the kind of the Fridays. What you'll see is in the black is those elevations. That we give them an hour of self-administration. It reduces their threshold. So it worked in these two other cohorts. When they were switched, so there was a week in which we switched them to only 1 hour, their thresholds did not go up, but self-administration for an hour didn't really do anything either to their reward thresholds. And then finally, we switched this group to heroin for 11-hour access, like the original oxycodone, just to see if it generalized to another opioid. And sure enough, it drove thresholds up. This is, again, the end of the week response. And then if you give them access to heroin for an hour, it drives the thresholds back down again. So that part is kind of consistent with what the model should be doing, which is that they're treating themselves, their dysphoria, by taking drug. So, and the other thing is that they're not broken. So we did another study too. We did acute administration of oxycodone, thinking, well, you don't get the dose right if we do investigator administered. We could not drive their thresholds down below baseline while they are continuing self-administering, even on those Mondays. So one of the fears you might have from behavioral models, a longitudinal study, I think it was like 12 and 13 weeks by the time we got to this. They've been doing it for a long time. Maybe the model's just broken, right? You've sort of burned out their brain and it's not functional. But if you discontinue them from drug access for 2 weeks or 3 weeks, challenge them with meth and then oxycodone acutely, they're not broken, right? Once they've recovered from self-administration pattern, acute administration of these drugs, a stimulant or oxycodone, will decrease their reward thresholds. So they have recovered. So whatever that Monday effect was, is probably real, right? It's not the assay was somehow broken, the behavior was something weird going on. It's maybe really true. So our best understanding of what might be going on is that if these are related, the failure to go down on Monday and the fact that that was the same time point when they were actually taking more drug, they were ratcheting upwards. If we bring these things together, what's possible is that what's important to the animal is the delta, the change, right? So on a Friday, when their reward thresholds are elevated, give them an hour of self-administration, their reward thresholds change in the better direction, they feel less dysphoric, they probably notice that. Whereas for whatever reason, if that's not happening on a Monday, the delta is not changing, so they're going to maybe take more drug trying to get an effect. Something along those lines is our best kind of hypothesis to go forward with about why we observe that and how we can fit these two pieces of information together. Oh, good. So I'm going to summarize a little bit. So in response to the opioid crisis, again, as I said at the top, I like to at least be somewhat informed in my research about what I think are relevant things for the human condition, for medical health, and the existing questions that might be out there. Vaccines are promising. We've known that for decades now in terms of biological efficacy. But I think we need to turn our sights onto what I call psychological efficacy to find out this weenie effect. So I didn't really talk about this much, but this sort of cottage industry has been driven by chemists who think about silver bullets in drug development. They think if we just make a better vaccine, we're going to get higher titers. You won't be able to oversaturate the sponge, if you will, like I showed you in some of the first data with changing the dose from 1 to 2 milligrams per kilogram. And if we just get the right vaccine, we'll have the silver bullet, it'll have ideal antibody titers, they'll be persisting for long periods of time. If you think back to the human trials, they'll be more consistent across individuals. That's what we need to do. But we've been at this since '74, and everything comes out the same, which is that you get kind of a weenie effect. And one of my colleagues who's peripherally involved, or pretty involved in this for a while, snarkily pointed out to me that if you look at all the different vaccines that have been made, the ones that work, the ones that survive this drug discovery effort, are the ones that look most like the parent drug coupled to an immunogenic protein. So all the flailing around about what the drug-like molecule is supposed to look like isn't as effective as just using the parent drug. That may not be completely true, but there's some truth to that. So they have a partial effect, which, by the way, small molecules do too. The treatment for drug abuse trials, small molecules, they have partial effect until you get to doses that have adverse side effects. So it's actually not that unusual. But my point is that we have to admit that this weenie effect, this small effect, whether it be 20%, 25%, depending what your endpoint is, that's what we have to work with. What we should be finding out from preclinical models is what can this tell us about how it might be applied to the human condition. Cannabis, THC, we're not as far along as we should be, but in these sort of environmental proposals that we should be using medical marijuana to compensate for pain control, we should be using marijuana to treat oxycodone addiction or opioid addictions. We need to be aware that at least so far, the evidence seems to suggest that what you're doing is you're boosting up the efficacy of a unit dose of the opioid. And the outcome of that is not clear yet. It could be beneficial. There's reason to think it could be very beneficial, and there's reason to be concerned. Okay, what did the other studies tell us about the human condition? Well, first of all, from a very practical standpoint, if any of you has ever had an older relative that's been prescribed an opioid, many of their responses that they're very worried about using them, they're very worried about addiction, they're worried about dependence. And so what do? Especially my father-in-law. Tough it out, right? I'm not going to take it. Okay, I'm going to take one. That may not be a good idea. We're kind of a far disconnect from, as I said at the top, we're not using paid models. And that's the next frontier that we need to look at in the animal models. But let's suppose it holds out to be true. Our kind of data would suggest that this is a very clear clinical prescription. Just take them on schedule, buddy, right? Don't take breaks. Don't try to tough it out. Just take them the way you've been prescribed. And if you go back to that epidemiology, we do need to reduce the length of prescriptions, sure. But one implication for our data is that they should just be taken on schedule and not have these breaks, because you might have some weird things going on in terms of your neurobiology if you do that. The behavioral data, the kappa opioid manipulations, the electrophysiology all point to dysphoria as a key driver and kind of reinforce this negative reinforcement hypothesis in a novel way, I think. But then, as I was just summing up with, the intracranial cell stimulation reward data are inconsistent. And so we don't know why. There's a couple of, kind of ugly conclusions, which is maybe it's just that our hypothesis about what ICSS is doing in terms of an index of the dysphoria related to drug taking and drug discontinuation, it might just be wrong, right? So we may just need to know more about what ICSS is actually telling us. The other potential nasty takeaway is that the behavioral pattern is not driven by growing dysphoria. And so I kind of hand-waved a little bit about how we're trying to fit those things together, but obviously we need a lot more investigation to kind of firm up why these two things are at apparent odds in some ways. Thank you for your attention. As I mentioned, Jacques did a lot of this work with Yannabel. Kevin chipped in. Electrophysiology was Marissa's lab with Dean. And then the vaccines in a sort of long-standing and very heroic collaboration with Candy. And then finally, NIDA for my funding. And thank you both for hiring me and for Neil inviting me to give this talk.
And thank you for giving this talk. A wonderful summary of your work, and we're lucky to have you in our department and to bring this work for us. I'd like to open this up for some questions. I'm going to start with trying to steer this towards the clinical implications. One of in the vaccine work, your data look a lot like what we see with many treatments in psychiatry, which is that there's some populations that get benefit and others don't, and when you put them both together, you have no significance. And so the question is, are there immune markers or other biomarkers that we can get either at baseline or after the first, vaccine administration that are going to tell us that we have a sensitive population that we could use then to steer our clinicians to either use or not use this type of a vaccination approach?
So obviously this is a long, again, with the two failed trials and there's sort of, aborted attempts to get another trial going, it's going to be a long time. Yes, people are very concerned about that. They're working with adjuvants and other things to try to tighten up this distribution, so to speak. And as I mentioned, they're trying to work on different immunogens or different designs for the vaccine to try to get this more consistent. Again, there's some preclinical work here that, can you use multiple vaccines? Like you have multiple vaccines for flu to get different strains. It might be that you pick up different individuals. If you give them like 3, they maybe not be developing the same. But you can see kind of in the beginning there, right, in the beginning of the inoculation series, that, yeah, some of the individuals take off earlier, right? So
Sure.
maybe about 8 weeks, you'd be detecting those who are going to be high antibody responders. But I don't know enough about basically antibody and B cell immunology, which another thing I say about this business is that that's the person that's been missing from this relationship. It's been chemists developing vaccines. It's been behavioral people evaluating the efficacy. And the role of actual immunological B cell antigen people has been missing in this course of, some of them came on lately, but they haven't been there. So yeah, people are working on it. But I think there's, it's very daunting for when somebody sees a failed trial for something that people don't want to develop medications for in the first place.
Sure, and that's why we're in the situation we're in psychiatry, which is a lot of drug companies are bailing because
Yeah.
of that exact issue. But the smart people, and I'm sure we have them here in our department, because one thing we do have here as an interest in the immune response is to figure out who are those folks, and if it's a way we can tell them.
Yeah, and it's crazy making for someone who's not in clinical trials, right? Because I kind of understand intellectually you can't do these trials and then do this post hoc analysis, right? That's what's not allowed. But you look at that and you say, well, but if people hadn't been taking the medication, you'd kick them out, right? And to me, that's basically what this is. They hadn't developed an antibody response. They're the same as if they hadn't been compliant with the med, right?
Okay, I'll stop dominating the situation here. And question back here.
So, you want the microphone, Igor?
It's Abe.
Yeah, okay, Igor. I'm gonna, and then I'll bring you the microphone, okay, to make up for. I thought that in the third part, Mike, you were going to tell me that you wanted to have either a compound that would hit both mu and kappa, or that you wanted to co-administer mu and kappa, so that you would be giving people a drug that wouldn't throw them into these troughs and therefore drive more self-administration. But you never said that.
Because you might have noticed my approach to that was not therapeutic, but using that tool as a tool to probe it. But you're not.
I'm now asking you what you believe. Yeah. Is that must be something you've thought about or not?
I'm obsessed by the paradox that we have with the ICSS right now. And so I've been trying to think about tools we could use to get at that as opposed to trying to find a medication.
Tell them what you really think about it. I've given away the microphone.
Thanks. Really great presentation. Appreciate it. On the vaccine story, you sort of mentioned the business about duration that the vaccine works. Is there anything more about that? And secondly, are there any concerns of autoimmune or other processes being triggered by these kinds of
So far, at least,
vaccinations?
especially in preclinical trials and in those few human trials and the phase 1s leading up to them, there's been no evidence of some kind of immune storm or some kind of implication that these are unsafe. So as far as the vaccines go, they seem to be fairly well tolerated systemically. What you're kind of getting at is, and one thing I kind of elided over, didn't make clear, is one of the biggest mistakes they ever made in this business was calling these things vaccines instead of immunotherapy or something of the nature of that, because people think of them as being silver bullets like we have vaccination for microbes, right? And they just don't work that way. Titer subsides if you stop boosting, right? And this is what you're asking, I think. So of course, maintaining titer is one of the goals, but unlike a microbe, so in a microbial vaccination, titer also subsides, and what wins is that you've got an insult that has a time course to develop in your body, and you're giving your body time to generate these antibodies more quickly that recognize the microbes and chew them up and get rid of them, right? That doesn't work for drug abuse because the taking of drug is a ballistic event. It's not like you're out racing it, right? So you have to maintain high titers. It's not a memory issue. So yeah, that is absolutely a concern. In terms of, like I said, I like to think about what are the implications for how can we use this thing that doesn't work as well as we might wish. Well, we know dang well that drug abuse is an adolescent disorder. So if you have to do something like keep boosting them every 6 months or something or whatever it might be, keep titers up, if you've only got a 4-year window with a teen, that's actually pretty accomplishable, right? Or if you're in a discontinuation of it, this is what people think of as the main target is people who are addicts and in cessation. It's going to be an adjunctive treatment. Again, it's not so daunting to think about maintaining antibody titer with retreating them for short intervals of time. So it may possibly be that, again, we have to accept that it's only a partial solution.
Okay, thank you.
Other questions?
Hi, thanks. Great talk. So if they're if you give the vaccine and they escalate their level of cocaine intake or something to try to get it into the brain,
Yeah.
is it dangerous to have those high levels in the plasma?
In the periphery.
I mean, aren't
Yeah,
in the
effects on
periphery?
other
Yeah.
compartments.
So are you? On the heart and
Yeah,
is that a worry?
so when I came into this, what I realized right away is that I'm a behavioral pharmacologist and I'm an idiot, and I think of basically 2 compartments: the blood and the brain, right? But of course, most of this drug goes elsewhere. Now, of course, it's going to have the same kind of efficacy, right? If you're retaining the bloodstream, it's not getting into the other tissues, not getting into the liver, it's not getting into the heart. So you are having you're presumably having a reductive effect. People have not focused on that. I'm not really familiar with these other systemic insults, but I would assume, again, based on the mechanism of action, that if you're retaining more of the drug in the bloodstream and it's being eliminated, you're protecting other compartments just like you're protecting the brain. I would assume. There may be different kinetics, right, and different distribution kinetics, but should be having similar effects.
It's going to have to get out of your system somehow, and you think maybe at the end of that process there could be some saturation. But other questions? Oh, there.
Thank you. Great talk. I'm just wondering, is there any evidence that THC could be directly affecting like the expression of the opioid receptors or where they're located on the cells or in the lipid membrane? Or
I don't know.
Okay. It affects.
Would you like to look into that for us?
Yeah, I would actually. Thank you. I'll talk to Jacques.
Yes, you should.
Mike, great talk. I was wondering about the third part of your talk, the ratcheting pattern that you've been describing. Do you think of it as a unique to oxycodone? Do you think it happens across opioid drugs or also psychostimulants?
So there's a history of this pattern. Like I said, so one of the reasons we saw this, of course, because I'm a nice PI and I just run things 5 days a week. So even with stimulants and even in short access, you'll see hints of this, right? Maybe it's not statistically resolvable. You'll see this deprivation effect. There's a whole cottage industry in the alcohol research that dates back decades of this so-called deprivation effect, right? So it's not that strange to see something like this for drug abuse to get this kind of increased drug taking. Now, in terms of prior opioid long-access models, I can only tell you maybe. So unfortunately, which I alluded to, or maybe I didn't allude to, but if you look at some of the Kubian heroin long-access things, you'll see animals will run 5 to 7 days a week. And the conventional wisdom when I started doing this was that, well, sometimes you need to give them breaks even more than 2 days because the animals start, frankly, chewing their fingers and self-injurious behavior. And so since the goal in those studies was to reach terminal, stable, high levels of intake and then look at what the consequences were, they weren't so concerned about these niceties. So if you squint sideways at some of those behavioral patterns in those heroin escalation studies, you can see things that look suspiciously like this. Now, they may have been attenuated because maybe only some of the distribution was given breaks and some wasn't. You don't know where the lineup is in terms of what time, but you can kind of see it there in some cases. And I just haven't, demanded the Excel sheets from Joel to see, if that's really what that was. So it may not be unique to oxycodone. On the other hand, it could be that whatever the pharmacokinetics are of this particular drug, maybe it's just that we saw it for this drug because of the specific properties of this particular drug. And if you titrated the discontinuations and the access intervals just right, you might be able to see it for other ones as well. We really don't know.
Carla?
Hello, I just have a thought kind of on this same topic, sort of kind of related. And this might be getting a little granular, but what's interesting, I think, in clinical observations is how quickly people metabolize opioids, and it's highly variable. So like in methadone populations, we do peak and trough levels, and patients on the same dose can have wildly different peak and trough levels. And when you look at heroin users in the real world, you have people who can use once every day and not experience too much withdrawal, and then you have people who have severe dysphoria and withdrawal and self-administer 5, 6, 7 times a day, right? And so it's really quite variable, at least amongst humans. And I chalk a lot of that up to how quickly they metabolize the opioids and how perhaps their drug levels are higher or lower. And I think there's some evidence to support that, in addition to what you're talking about, other explanations for this. But I'm just curious, in animal models, how do you look at the blood levels, or do you look at the levels of opioids and see how that maps onto these different time courses that you're looking at? Because I think that would be also something that that is at play at that same sort of thing when you look at the development of the dysphoria.
Generically, the half-life of oxy in rats is, on the order of a day. Not like, you're not talking about the fact that you've got drugs still out there at 60 hours of discontinuation. And there's not a lot of genetic variation. We don't do the PK. You might want to hammer Abe and Olivier, who are doing different rat strains that may potentially vary in those properties. We can, yeah. Possibly. I mean, so we use the same beastie, right? So they're going to have some variation, but it's going to be constrained, presumably. With stars, yeah.
Yep. Outbred rats are good for variability. Any other questions? I've got try to bring it back to the clinical issue here. First, no one's commented on the fact that you've identified a model for the TGIF effect, which is kind of interesting.
It's actually more reinforcing on Friday, right?
That's right.
Yeah.
Yes, absolutely. Drugs are more reinforcing on Friday, and that's why everybody's popping on Facebook. Yeah, everyone's popping on Facebook, and they're in that other bar, the second bar on the Friday stuff. Yeah. But something that wasn't discussed about the new opioid crisis and the OxyContin, et cetera, is that, and one of the reasons it's become such an issue is not just that people are taking more of it, but because people are dying. Mm-hmm. And that's a hard endpoint to study in your animals. But I'll channel Matt Klein back there because he wanted to ask this question, and so I'll ask it for him. So the fact that people are dying, and presumably some of that is a function of the potency of these drugs and respiratory depression and things like that, but some of it may also relate to suicidality that's increased in this group, more so with the OxyContin and other related drugs than with the heroin population. I'm wondering, has anyone studied models in your animals or the types of animals that are chronically administering OxyContin to look for models of depression or things that might be informative to us about suicide?
Well, so I don't. We have models that we say anxiety-like behavior, depressive-like behavior. Yes, of course, as you know. And those have been important to suggest that in these long access models that animals are dependent, right? Because if you give them discontinuation, you trigger species-typical discontinuation syndromes, including on anxiety-like behaviors, depressive-like behaviors. So yeah, that's true. We haven't really looked at that. We did a little bit of work with this to show that they were, what did we do with oxycodone? Did we do it Clusmaze, or we did, yeah, so something. We haven't worked on that very hard. Yeah.
Because obviously, what's making this a national crisis is not just that more people are taking OxyContin, but
it's the deaths. Like I started out, that's what got people's attention. I think, you really, the first thing you said seems to be right, which is that they're just highly potent. People don't know what their dosing is. And it's not just people intentionally trying to get opioids. Fentanyl's turning up in the cocaine supply. So a year or two ago in PB, where I live, we had 3 deaths from cocaine contaminated with fentanyl. So, they're not intentional necessarily. But again, you're right that there is this maybe potentially, suicidal / adjacent population that maybe is careless, I guess. But no, I don't know about animal models.
Be a great thing to study. Any? Yeah.
Well, you heard, I don't know if you were here for Coop's talk, but one of his many new buzzwords is this deaths of despair, right? That there's this increase in suicides in middle-aged people who otherwise seem like they're good. This question about drug abuse. So yeah, I mean, there's interest. Great.
Well, so first I want to thank you again for this outstanding presentation. And as Susan Tabor could tell you, that one of the traditions we have in this department is that if we bring you on board, you have to wear a silly hat. And so this is the hat of UC San Diego Department of Psychiatry. I want you to wear it in good health.
When I saw Igor give one of these to Yasmin Hurd, I was like, how do I get my hands on one of those? That's great.
Thanks very much.
Yeah, thanks.